Welcome to the Chapter 7, Part 1 lecture on passive transport. You should use the information in this lecture to complete the Chapter 7, Part 1 guided notes, which of course you should complete before you attend class. So first you need to understand a little bit about the structure and the function of the cell membrane. Here we have the cell membrane, and what this diagram is showing is that the cell membrane does in fact surround the entire cell. Now here it's shown as a sphere, and the cell isn't always spherical. Because this membrane is in fact a liquid, it can take on different shapes depending on the cytoskeleton that's inside the cell or a cell wall on the outside. So cells can have different shapes. They can be rectangular. Some of them, like nerve cells, can even be kind of star-shaped, which is kind of fun and weird. Or they can be round like the sky. The cell membrane is made of phospholipids and other components that we'll get into, but it does separate the external environment from the internal environment. Cell membranes are semi-permeable largely because of the structure of those little phospholipids. So let's take another look at that. Just to remind you, the heads of the phospholipids contain a phosphate group, and that makes them very, very polar, while the tails contain methyl groups, which makes them very, very nonpolar. When you put these phospholipids together in a larger membrane, you end up with the very outside of the membrane and the very inside of the membrane being very polar, with this big, thick nonpolar layer in the middle. Now really, it's the nonpolar layer in the middle that kind of helps decide who can come in and who can go out. So that sort of begs the question, which molecules can do this? Which ones can cross by themselves and which ones can't? In general, small molecules and molecules that do not have charges can pass through the membrane by themselves. Here, for instance, we have a little nonpolar molecule. Because the center of this membrane with all those tails is nonpolar, and nonpolars like to mix, this little molecule should be able to cross that membrane by itself. It's also pretty small, so that helps it squeeze between the phospholipids and get across that membrane. Again, in general, large molecules and charged molecules are not going to be able to pass across the membrane by themselves. Well, why is that? Well, here we have some little ions, for instance. Ions have charges, and because they have charges, they don't like to mix with this big nonpolar layer in the middle, which has no charges. Things with charges tend to like to mix with other things with charges, but they don't like non-charged molecules. So they're going to have trouble getting through by themselves. Also, molecules that are very large, like this big old macromolecule over here, is going to have trouble squeezing its way between those phospholipids, so it's not going to be able to cross by itself. Incidentally, take a look at this macromolecule. The shape of it should look sort of familiar to you. That molecule is most likely a protein. There are exceptions to these rules, and water is a great one. Remember that water is that small, sort of rebellious little molecule that just kind of does what it feels like. Even though water is very polar and has partial charges around it, it's also pretty small. So water is one of those tricky molecules that can usually pass through a membrane by itself. It's able to sneak through that phospholipid bilayer by itself and go either direction, into or out of a cell. That makes it a little bit trickier for cells to control, but it has some methods for that. The general motion of molecules is known as Brownian motion. This lecture and your pre-lab for Lab 5 will just refer to this movement as just movement by itself. Molecules are constantly in motion in every direction. Now inside a gas, those molecules will have the most kinetic energy, so they will have the most range of motion. They're moving around a lot. Inside a liquid, they have a little less kinetic energy, so they move a little less. And inside a solid, they have the least amount of kinetic energy, so they can't move very much. But even inside a solid, those molecules are going to be vibrating slightly. So remember, when you do your pre-lab and when you answer questions about movement, if it's just movement by itself, molecules are always moving in every direction as much as they can. Now, the term net movement is a little bit different than just the term movement by itself. It's different than plain old Brownian motion. Net movement implies that there's some sort of an imbalance in the system, and because of this imbalance, molecules are moving in a specific direction for a specific reason. What kind of an imbalance am I talking about here? Well, here, for instance, we have an imbalance in concentration. We have a high concentration of these molecules on one side of this barrier and a low concentration on the other side. So that makes the molecules want to move from high to low. There's some difference that's making it move in a direction. When we have a difference in concentration or in pressure in two different areas, we say that a gradient exists. And when gradients exist, there will be net movement or movement in a specific direction. So if you're asked a question on your pre-lab or in class about the net movement of molecules, the answer is always going to be something like top to bottom, inside to outside, right to left, something like that, in a specific direction. In this chapter, we're mainly going to be dealing with concentration gradients and how cells use concentration to make molecules move where they want to go. There's a couple of things to remember about concentrations. First, they can be measured in percentage or molarity, and we'll talk about molarity in a moment. When you're dealing with percentages, it's important to remember that the percentage of the solute and the solvent is always going to add up to 100%. So here, for instance, if we have a solution that contains 10% salt, we know automatically that it must contain 90% water, because 90 plus 10 is 100%. The other thing to remember about percentages is the higher the percentage, the more of that component is going to be present in the solution. So for instance, a solution with 70% solute is going to have a lot more solute than something with 10%. Another way to represent concentration is, of course, good old molarity. And there's really just a couple of things you need to know about molarity for this chapter. First, you need to remember that the higher the molarity of a solution, the higher the amount of solute it has. So a 1 molar salt solution has more salt in it than a 0.5 molar solution. The other thing you need to remember is that the amount of solute and the amount of solvent within a solution are indirectly related. So the higher the amount of solute, the less water you're going to have in your solution. That means that a 1 molar salt solution will have more solute, but less water than a 0.5 molar solution. Gradients exist anytime you have two areas that differ in some characteristic. For instance, here we have a concentration gradient. We have an area that has a high amount of solute here and a low amount of solute here, so there's a difference in the solute concentration. Therefore, we would say that a concentration gradient exists between these two areas. Now we have a few vocabulary terms that you need to know that have to do with concentration gradients. If molecules are moving down or with a gradient, that means that they are moving from high to low concentration areas. So for instance, if we have a molecule moving from this side of the solution to this side, from high to low, we say it's moving down or with the gradient. On the other hand, if we have a molecule that's moving up or against the gradient, that means it's moving in the opposite direction, from low to high concentration. So for instance, if we have this little molecule over here and it's moving from an area of low concentration and getting more crowded to an area of high concentration, we would say it is moving up or against the concentration gradient. Which of these motions do molecules prefer? Well, in general, molecules like to move down or with the concentration gradient when they can. In other words, they like to move from where they're all crowded up to where they're all spread out. Think about this like getting on an elevator. If you got on an elevator and only one other person was on the elevator and they stood really, really close to you, you would feel really uncomfortable. Molecules are like that too. They like to spread themselves out. They like to have some social space. So in general, they try to move from areas where they're crowded to areas where they are not crowded. So now that you understand a little bit more about how molecules like to move, let's circle back around and apply this to living cells. How do living cells control which molecules move from the intracellular environment to extracellular and vice versa? Well, there's two major types of transport that cells like to use. The first type of transport is known as passive transport. And passive transport allows molecules to move the way that they normally would like to. They go where they want to go. Remember that molecules generally like to move from areas of high concentration to low concentration, down the concentration gradient. So here we see that. We see that these little triangles are crowded on the extracellular side, and they are less crowded inside, so they want to flow through the membrane and spread themselves out, moving from high to low concentration. Passive transport is the preferred type of transport by cells because this requires less energy. These molecules have kinetic energy, and they can use that to move where they want to go. But this type of transport requires no extra ATP energy on behalf of the cell. So because the molecules are moving the way they want to anyway, the cell doesn't have to expend any extra energy. So they prefer this method. Another method of transport is known as active transport or active processes. Active processes are the opposite of passive processes. In active processes, molecules are forced to move against their will, against or up the gradient, where they don't want to go. For instance, here we see a low concentration area of diamonds in the extracellular environment, and you can see that there's a higher concentration inside the cell. The cell is having to force this little diamond-shaped molecule through that membrane and across into the intracellular environment. It's making it more crowded, so it doesn't want to go that way. Now, because molecules are being forced to move against their will, against the gradient, the cell does have to expend some energy. And this energy is always going to be in the form of ATP. Here you can see an ATP molecule being broken down for energy to power this reaction.